Okay, let me actually get started. Good afternoon, good morning, good evening, whenever you are. So I would like to welcome everyone to our theoretical and computational biophysics seminar today. Today's guest is Professor Peter Tieleman from University of Calgary. Before I introduce Peter, I remind everyone to please turn off your microphones. You are encouraged to keep your cameras on, so that gives a little bit more interactive feel to the seminar. It's like we are sitting in the Beckman Tower room for the seminar and we are looking the seminar speaker in the eye. But please turn off the microphone. If you have any questions, try to keep them toward for the end of the seminar when we have the Q&A. If there is a real burning question, then I hope Peter doesn't mind to be interrupted once or twice maximum during the talk. But again, you can post them in the chat box or in the Q&A box, whatever you see, and then I try to moderate the questions and answers toward the end. So with that, it's a great pleasure for me to introduce our great friend and colleague, Professor Peter Tieleman from University of Calgary. Peter was trained initially as in chemistry and philosophy. I didn't know that part, I have to say, at University of Groningen in the Netherlands, where he also did his PhD in chemistry under the supervision of famous, one of the pioneers of molecular simulation in Europe, Hermann Berendsen, where he did his PhD there. And then after that, he received a fellowship from EMBO, during which he worked with Mark Sansom at Oxford. And then immediately after that, he joined the faculty at University of Calgary in 2000, where he is currently a full professor and the Canada Research Chair in molecular simulation at the Department of Biological Sciences. So Peter and our group, we have a lot in common. So we are interested in molecular simulation methods, software, and a simulation of membrane-associated systems, processes, and phenomena. So it's really nice to have him every now and then to get an update. He has received quite a few awards during his career, and he has been very productive. His H-index is H84 with more than 30,000 citations. And he is also involved in development of software, actually. So he is one of the contributors to GROMACS and also the Martini coarse grain force field that many of us use. So he has been kind enough to also serve on our advisory board for several years. I was just telling him that we missed it this year. Hopefully next year we can resume the activity. So it's very nice to have you here, Peter. Thank you very much for accepting our invitation and to agree to give us a talk. And we look forward to your presentation. Please.
Thanks. It's pretty amazing to look at the list of attendees and recognize a lot of people, including people I haven't seen in a long time. Hi, Luca, Mark, Robert, others. So in this COVID time, I've read different stories about the effects of a year of stress. And one of them is people end up being stressed and have difficulty focusing, which I think is something I suffer from at the moment. So compared to other talks I've given, this actually will touch on a lot of different things, but all pretty briefly, just showing the kind of things that we're working on and the kind of things that I think that are working at the moment in terms of simulations applied to membranes. There's a little bit of a historical part to this, and many of you were part of that history. Certainly, people like Amat and Barry go back the same time almost in membrane simulations as I do. And then there are a couple of things about Martini 3, which is new. I promised Amat lipid-protein interactions in Martini 3, but that was a bit premature. I'm going to talk about simulations that use mostly Martini 2, that use Anton, that use atomistic methods. And then I'll talk a little bit about the capabilities of Martini 3. And there are some people on this webinar that actually know quite a lot about Martini 3. In some cases, probably more than I do. So in general, we are interested in membranes. This is a textbook picture of a membrane, and this is probably too simplistic. This is now 20 years old, and it should be more crowded, it should be more diverse, and there should be more proteins, more interactions with things outside the membrane. But it shows some idea of the complexity. This is our computer model from 1996, around the time I started working on this, and that's a few years after Herman Berensen and Klaus Schulten started working on this for the first time, making models of phospholipids. I think all of you, or almost all of you, will have a background in simulations. So I introduced this for a number of different reasons. I use this slide a lot. One is just to describe molecular dynamics. You describe the interactions using classical potentials, and then you calculate how things move based on equations of motion, generate an ensemble, and with statistical mechanics, you can calculate thermodynamic and kinetic properties from the simulation. That's basically what we're doing in the rest of this talk, and a lot of us, or most of us, are doing. The second point I want to make with this is the time scale of motions. This is a movie from 1996, and it was kind of state of the art back then, but the time scale was half a nanosecond or a nanosecond, which took a long time to run on the power challenge machine that we had in Groningen. It was pretty rare at the time to have a machine of that power in an individual university. So for a single component liquid crystalline bilayer, nanoseconds were a pretty good time scale. I mean, you can do a little bit better. If I would do this now, I would ask for 50 nanoseconds, but that's not wildly better. That's a little bit more statistics. But that's enough to get data on individual lipid components. And then we were kind of stuck in that for a long time, because if you look at the timescales in actual bilayers or in membranes, they're massively longer. And this is an example from almost 20 years later. This is a paper with Yi Wang, many of whom will know her. She was a member of group in Urbana as well, and many of you are still working with her. So we did simulations on mixtures that were basically atomistic model mixtures for eukaryotic membranes, where the simplest two-component model is PLPC cholesterol, and the simplest bacterial two-component mixture is PLPG, PLPE. And in order to get enough statistics on that, you need microseconds. So this is 3-4 orders of magnitude longer than my previous movie. And that's 20 years later, which approximately matches our increase in computer power, factor of 10 every 5 years. And then both Chromax and NAMD have become more efficient. So even on the same machines, they would be a few times faster than they were for older generations. Still, you need microseconds to look at binary mixtures at a liquid crystalline, still a liquid phase. And this becomes longer if you add a lot of cholesterol, or you go to phases that have gel-like domains where motions slow down and are almost impossible to simulate atomistically at the moment. This was Anton 7-8 years ago. So now a few microseconds on these systems is simple on a single GPU. But again, we're almost 10 years further now. So that's 2 orders of magnitude on computer power. This is from a cover of a chemical review issue. Some of you contributed to as well, including Amat with a killer review. This is our current most complex membrane model. This is from Theo Jan Maring's group. We have similar things. This is using Martini, but this is the kind of complexity that actually is approaching the cartoons I started with, and is actually more realistic in terms of density, in terms of volume, in terms of interactions with other components of the cell. So we are reaching scales like this. I have no slides about this, but was this big collaboration that Klaus started a long time ago, and many of people in Urbana worked on the chromatophore, 130 million atoms. That's an atomistic model with this kind of complexity and kind of density. That requires the biggest computers available at the moment. But with Martini, you can get sampling on systems like this to some degree. So you mentioned Martini before. It's a coarse-grained model, and we spent a lot of time working on this since you and Jan started with this almost 20 years ago. So the basic idea is to map individual atoms to beads that are no longer recognizable as atoms, but have a lower resolution, which means it's much cheaper to simulate. And the effect is quite staggering. It's 2 to 3 orders of magnitude faster than atomistic simulations. You lose chemical specificity, you lose detailed interactions, there are other things you miss, but the calculations are much cheaper, and for many cases, you continue to have enough detail for a lot of interesting processes. And in particular, lipids have been, I think, a success story with Martini. So on the left is a figure from the original Martini paper introducing the mapping for phospholipids and cholesterol. On the right is a figure from Luca Monticelli's paper, who made the first protein model when he was in my group, collaborating with people in Groningen and in Michigan on mapping Martini to amino acids. And if you look at this, this looks like some of the residues are the same, like isoleucine and leucine looks very similar, but if you look at the numbers, they actually are slightly different. But this is kind of the level of resolution in Martini 2, and then some refinements in 2.1 and 2.2 with the same mapping for modeling proteins. So there's a new paper coming out in Nature Methods soon on Martini 3, and I'll have a few more slides towards the end about that. But that's a deviation in some ways, although the philosophy is still the same, that massively increased the capabilities of Martini. We've also worked on trying to link different levels of resolution, and that's not exactly a solved problem to do it in an automated way. And unfortunately, it depends on software engineering and clever algorithms. I would like to have a way of automatically going between different levels of resolution. For a while, this seemed to work. At the moment, it seems to be a bit more finicky than we would like. But in principle, you can go from very coarse lipid models like this one from Marcus de Cerno, where you have 3 beats per lipid, to Martini, to atomistic lipids or united atom lipids. And going back is easier because you just have to average over degrees of freedom. Going from here to here is harder because you have to add information. And that information is available because you have topologies, you have the chemistry. A lot of it is practical. How do you prevent chains from sticking through rings in simulations? But ideally, you would be able to quickly interconverge from different levels of resolution. Hi, just saying hi. Okay, I was looking at the chat. Maybe I should not do that. Okay, let's go on. So with Martini, you can make very complex membrane models. This too is from 7-8 years ago. This is from a paper by Helge Ingolfsen and Jax. It was one of the most complicated membrane models around, and it still is one of the more complicated models. I think most people have to actually try to go back in complexity to something less complex because with this many components, it's difficult to make sense of what's happening. This was a very cool paper that looked at an asymmetric bilayer, introducing asymmetry, which is currently a hot topic in membrane biophysics, and composition with more than 60 different lipid types. If you look at cell biology papers or lipidomics papers, people identify up to 1,000 lipid types in many cells. But the components of the distribution of these is such that the number of lipids of more than 1% or so abundance are rare. So 60 covers a lot of these lipids. I won't go into this story anymore, but we use this model to look at lipid-protein interactions. And the initial question we asked is very simple. What happens if you stick proteins into a lipid bilayer? And until about this time, people looked at interactions between specific lipids, like cholesterol and GPCRs, or poly-unsaturated chains with rhodopsin, also a GPCR, or with ion channels. But with Martini, the timescale of tens of microseconds, and this kind of complexity in your lipid membrane, where you need tens of microseconds to get sampling over diffusion, you can actually look at what happens if you put a protein in a complex, more biologically relevant lipid mixture. And some of the questions were initially very simple. Like from a technical point of view, how long does it take to get equilibration of these distributions? Because what's going to happen, you start with an equilibrated bilayer, you add a protein, and now the system is out of equilibrium because the protein will affect the distribution of the lipids. Then after a certain amount of time, you should reach a stationary distribution of lipids, although lipids move in and out close to the protein. That gives you converged average properties of distribution in the presence of these proteins. From a simulation point of view, for the larger groups, you need tens of microseconds. For the smaller groups with less than 1%, sampling is much harder and you need much more. And we also had some estimates. If you would do this atomistically, you would probably need a factor of 4 or 5 longer, because in Martini, lipids move around faster than the timescale suggests. So 1 nanosecond of Martini for lipid diffusion is probably closer to 4 nanoseconds in physical time, although this is not a constant factor, and that's actually difficult in terms of mapping timescales. And what you see in this is initially a lot of, well, what we got out of this initially is a bunch of numbers. So you get concentrations of lipids close to the protein compared to bulk. And we've called this enrichment or depletion scores, where an enrichment means that there's a concentration of more than 1 times the bulk concentration, and depletion is less than 1. And one of the things you see immediately is that in this particular case, this is aquaporin, one of Amon's old favorites, although this is aquaporin 1, with poly-unsaturated lipids jumping on the protein. And there's a technical question here. It turns out that most of these poly-unsaturated chains are gangliosides, so they have carbohydrate head groups. And we weren't sure whether these interactions are real or not. We actually wrote the whole paper about this, and we still weren't sure. And now I'm trying to figure out if we can do experiments to measure this. Anyway, the point is, you see depletion enrichment of any lipid class around the protein. So the environment of the protein is never the same as the bulk concentrations. And you get properties like this from the simulations. You can calculate the concentration or the enrichment or depletion relative to the bulk concentration. And you find binding areas around aquaporin for different kinds of lipids that are enriched or depleted. Well, if one area is enriched, the other one must be depleted. There's some trivial component to this. And you also find binding sites for specific lipids. Most of the things that we were interested in, though, are not actually binding sites in the sense that this lipid binds to this protein and sticks, it has a binding affinity of a micromolar or whatever, and it's always there. What we're seeing in many cases is that there's a change in the total concentration without very strong binding, but this has a large effect on the overall structure of the membrane. You can look at the local thickness, you can calculate local curvatures, Gaussian and mean curvatures, and this is related to how these proteins might talk to each other in the membrane. So if you have a membrane with lots of proteins, and an individual protein has a strong effect on the local curvature, these effects add up, and people have looked at this. Marcus de Cerno has a very cool paper in Nature years ago that used a simpler model that showed that small effects add up to basically completely deforming the membrane. Greg Foth has papers on this. Jose Ferraldo Gomez has a cool paper on mitochondria cristae with ATPases that together bend the membrane in mitochondria. So I think this is an organizing principle in membranes that is quite important. It's not so easy to look at, but I think with these Martini large-scale simulations, you can. So we had a whole panel of proteins, and I actually didn't include the slide with all the proteins. The list is here: aquaporin, COX-1, DAT, EGFR. These were selected, or I selected these initially 6 years ago or so, based on available eukaryotic structures representing different classes of membrane proteins. And we looked at all of these, and we showed that the lipid environment around all of them is different from bulk, and also different from each other. So each protein has a unique lipid environment. And we call that a lipid fingerprint, which is, I think, a term that's being used in the literature more now. This paper is, I think, pretty widely read. The simulations also give you access to details. So this is P-glycoprotein, an ABC transporter. And this is basically mapping the Martini simulation, the curvature on a plane, showing the atomistic structure, although the simulation was Martini. And then you can actually look at correlations between residues on the protein and effects on the membrane. Or you can look at residues on the protein periods. P-glycoprotein in particular has a large following of thousands of people who write papers, thousands of papers a year on P-glycoprotein. So I don't actually remember all of these residues, but there's a correlation that you can look at between membrane deformation and function in some cases. So we did this for CFTR, where the EM structure, if you do simulations, actually has strange effects on the membrane, and the EM structure itself looks a bit wonky. It's still not clear whether that structure is wonky or whether there's actually something interesting mechanistically there. But this is the kind of thing the simulations can pick up, at least as something to follow up on. So in pig glycoprotein, we noticed the blue fuzz is pig glycoprotein. The colored stuff is a density map for cholesterol. So there's cholesterol around the protein that has a particular structure. But we also noticed that phospholipids actually have 3 well-defined binding sites in the cavity of pig glycoprotein. I thought that was pretty cool at the time because I did not really anticipate you could see this in a simulation other than if you put these lipids in there. But on the timescale of these simulations, you actually see lipids moving in and out, and you actually can find binding sites with quite a high degree of precision. We followed up on this. So now the gold fluff here is the protein, and all of these spheres are a time trajectory. So it goes from red 0 to blue 20 microseconds. This is Martini. And basically you follow the trajectory of a single lipid as it swims around through the whole simulation box and then enters the protein, and then it stays there. And this second graph is a similar graph, it just follows a different lipids initially in the cavity of the protein, and then it stays there for 10 microseconds and then it moves around. And when you look at this and you think back to my 1996 movie, in 100 picoseconds, lipids move from here to here. In this simulation, basically individual lipids go everywhere. And you have enough statistics to actually define entrance pathways or exit pathways and portals in pig glycoprotein simply by making densities of lipid molecules moving in and out. So this is a particular case where this lipid has a limited binding affinity. They interact, or they exchange quickly, but they're always there. So this density is very well defined. And that's a feature that's not so easy to find in other ways. We followed up on this 2018 paper with the 10 different proteins. And one way of doing that is by looking at other proteins. So this is a little bit trickier. This is GPCRs. These are not different families, but they're all GPCRs. And based on PhD students in my group who graduated a few months ago and is now at St. Jude's, did simulations of 28 different GPCRs, almost all of the available structures when he started doing this. They have different families. They come from different branches of the GPCR family, and some of them have been crystallized or structure determined in different states. And I'm not going to show any of the details here, but basically what you find is the same story as for the other proteins. They have different lipid environments, but now they're actually very close for GPCRs within the same family and different for GPCRs in different families. So even though different GPCRs with different substrates or different functions, if they're in the same branch of GPCRs, their lipid environment tends to be similar. We also find a large number of specific binding sites, and for GPCRs, there's a lot of literature on this. A lot of it is simulation literature, and I think in a lot of cases we don't know whether this is functionally important. But there are lots of papers that find cholesterol binding sites on GPCRs or PIP lipids bound to GPCRs. And there are a handful of structures. So we find most of these structures that are experimentally available in terms of lipid binding. And then we find a bunch of others that could be followed up on. This is an example. This is a family of 8 GPCRs that are related. And they all have cholesterol binding sites or binding environments that are in similar locations, even though the sequence identity is not very high, and even though there are no real cholesterol binding motifs. There's literature about crack motifs and CARC motifs, but those are really lame. And there are lots of false positives and false negatives. And also those are sequence motifs, whereas cholesterol tends to bind across different areas, not actually for adjacent residues in sequence. And there are some attempts at classifying those binding sites too, but that's not very specific, and it doesn't actually match what we're finding here. But evolutionary, you see binding sites for cholesterol in related GPCRs in related cases. I want to make a quick plug for an interactive analysis tool that Bayesian wrote. This is super cool, I think. So this paper was published in Biophysical Journal, and actually what we did what Bayesian did in this paper, he published the trajectory data together with a frozen version of this website. So you can actually go to the link here, which is also in the paper, and you can play with his density data, and you can do his analysis, or you can do additional analysis on the simulations that he did in terms of lipid-protein interactions, but we're not actually in the paper. And I think this is very nice. One of the problems with simulations, and we've talked about this for literally 2 decades, is that I generate terabytes of trajectories and I make 4 figures, or these days when you send stuff to Nature Communications, you make 1 figure with 9 panels, and that's the whole story. And the Martini 3 paper is like that. It has almost no information, except it had 200 pages of supplementary information that is harder to find. Anyway, point is, it's not easy to use simulation primary data if you have additional questions. This is a way of addressing that, and we actually had people interested in a specific GPCR that was in his set, asking questions that we didn't think of, but they could look at that using his website and his frozen version of this tool. This tool is very cool. So this is actually from a follow-up paper that we're going to submit to nucleic acids research that describes the interactive version of this website. So in this version, you can upload your own data and you can analyze it with the analysis tools that are part of this website. And then you can actually just use the graphs for whatever purpose you want, or you can share the data in the same way as he shared the GPCR data. These are some of the analysis it can do. It will basically look at interactions between all residues and all lipids, and you can classify and cluster these in different ways. It will calculate densities, curvatures, and thickness, and you can classify and visualize those in different ways. You can follow distributions, distances between groups you give, and there are a bunch of other things you can do with this website. It's pretty advanced. It's actually well beyond what I've ever looked at in terms of web software. It uses all of the coolest current visualization libraries like Bokeh. It has a database engine, and it basically is a JavaScript mixed Python system where you can also download the Python code. So if people want to play with this, I'd be very interested in and see what people can do with this, and if there are any suggestions on how to make this better or more interesting. So basically, I'm just finalizing the public website at the moment, but it has to be final by essentially this weekend, because that's the deadline for nucleic acids research. Okay, I want to talk about very briefly about a few applications of these lipid-protein interactions. This is from a recent paper by a student in Sergei Noskov's group that I was part of. This is on the potassium channel HERC. HERC is well known as a potassium channel because it tends to kill people as a side effect of drugs binding to it. They're not supposed to bind to it, and it causes arrhythmias. There's a lot of literature on this and a lot of controversies that I don't really care for. But one of the questions is very simple. A lot of the drugs that bind to this is a standard potassium channel, to my mind. If you're a potassium channel aficionado, there's more to say about it. But basically, this is the pore domain, 4 × 2 helices, and then there are 4 folded sensor domains. So the idea is that drugs that cause cardiac arrhythmia bind to this pore domain. But that's being disputed at the moment, at least for some drugs, because it looks like there are pathways for drugs to bind to the protein from the membrane. So rather than getting in the pore domain, it might bind to the pore domain from the outside, or it might bind to the folded sensor or some kind of interface between these different domains. And this story is part of that. So it's not looking at drugs, it's not even looking at biological lipids, but it's looking at ceramides. So these are lipids that occur biologically, but not in cases that are relevant for HERC. But they modulate the properties of this channel. And this channel can be interrogated by electrophysiology in great detail. We have a collaborator in Calgary, Hank Duff, who has spent his whole life working on this channel. And he can basically make any mutation you like, and he can basically measure any electrophysiology property you like. So you can look at the effects of ceramides added to HEC cells in experiments, and you can look at the effects of ceramides to mutant versions of HERC, where the mutants are based on simulations or on results from the simulations that suggest that ceramide binds to HERC in different cases. So I'll get back to that in one slide. This is a brief interlude. This is a different story. But this is also on HERC. And I'm just showing this because it uses MDFF, which is developed by the people in Urbana. So this is a simulation from Hanif Khan in my group and in Sergei's group on HERC, on an EM structure from Rob McKinnon. And that structure is controversial because from HERC and other potassium channels, we know there are lots of soul bridges that actually are not visible in the EM structure. And we played with MDFF in different environments, in vacuum, implicit solvent, in a generalized Born environment, and in an explicit lipid environment. And if you do that, you actually see a bunch of soul bridges form with almost no change in quality of fit to the EM density compared to the EM structure. So this is a distance plot, and the soul bridge distance cutoff is about 3 angstrom. So the EM structure has none of these 6 soul bridges, although one is kind of close. If you do a simulation with MDFF, constraining it to the density from the EM experiment, you actually see this formation of stable soul bridges. One of these was actually not seen before, or people didn't realize that was a soul bridge before. And Hank Duff, our HERC collaborator, actually made a reversal mutation and showed that this is actually a soul bridge. It doesn't have the same brutal effects on gating as the other soul bridges do. It's more subtle. But you can actually show functionally that this must be a soul bridge, because if you do the reversal mutation, the protein acts the same as in the native case. And if you disrupt the soul bridge, it acts weird. So this is MDFF. It was very cool. I'll probably use this again for other EM-related things. Now back to the ceramide story. Similar to the pictures I showed before of lipids binding to proteins, in that case, the random lipid mixture, this is more specific. This is ceramides asking, and we are adding ceramides in a low concentration, and we're asking where do they go. And by Martini simulations, you identify different binding sites on the protein. These are actually on the outside of the protein in the membrane, so they're not in the pore. But the functional effects of this look like the effects of some of the hydrophobic drugs that interact with HERC. Then there are additional simulations in the story using Anton. So we took Martini simulations to look at lipids partitioning, and then we took snapshots from the Martini simulations to start Anton simulations to get more detail on a 10 microsecond timescale. And then you can do some additional free energy calculations for a specific binding site for specific questions. So I don't want to go into too much detail about this story, but basically what you can show is that ceramides, adding ceramides, this 0.6 here, is kind of a weird one with a small, one chain is pretty short, actually will shift HERC to a closed state. And there's a whole functional story you can add to this. This is even vaguer. This is another story on KCNQ2, another cardiac channel. I don't even want to go through all of these slides or all of these pictures here, but this is a story where fatty acids modulate this channel. And this channel is involved in heart arrhythmias as well. And the hope is that poly-unsaturated fatty acid derivatives could be used as drugs. There is some evidence for this. There are some patents for Peter Larson, our collaborator on this in Miami, in this direction. But there's also some questions on how they actually work. And these fatty acids have opposite effects on different proteins. So it's going to be more complicated to turn this into something practical. But the biophysical story is relatively clear cut. You can actually do simulations with Martini initially on different PUFAs. You can turn them into Anton simulations to look at local binding in more detail. You can identify binding sites on these proteins where these PUFAs interact. You can mutate these binding sites. So now you have a sensor for PUFA binding experimentally, and you can show that experimentally these binding sites look realistic in the sense that if you change them, there are measurable electrophysiological properties that change. Okay, so now I'm moving to a couple of slides on Martini 3. Some of the highlights. Martini 3 has different particle sizes and more balanced interaction. So I showed the Martini 2 mapping of 4 to 1, 4 carbons or 4 non-hydrogen atoms to a single bead. That was standard, but then that turned out not to work for some things like benzene rings. There's no way to map those 4 to 1. So we had smaller types for benzene. And then that turned out to be not enough for nucleic acids, where you could not get base stacking unless you had a higher resolution. So ad hoc, we've had different sizes in Martini for basically since 2007 or 2008. But Martini 3 was designed from the ground up to have different particle sizes. So there actually are 3 different water models and 3 different carbon sizes and 3 different sizes for almost everything. So you can have a mixed resolution. The other thing that people have found increasingly with Martini 2 is that there's certain areas that are not very balanced. That's unavoidable if you start with a new force field. I'm still finding the same thing in optimistic force fields. And I think this is happening in 2 different areas. So one is that certain interactions are simply not exactly the right strength. But the other one is that Martini 2 turns out to not be able to distinguish between a lot of chemical groups that in organic chemistry are different from each other. So a lot of small organic molecules that are actually somewhat different map to the same Martini bead. So that loses some of the resolution. And there are certain areas of chemical space, of organic chemical space, that were barely covered by Martini 2 at all. So Martini 3 has more types, and it's also more flexible in terms of modifying types, with the aim of spanning all of chemical space. Martini 3 is also much better at parameterizing ions. Basically, Martini 2 had sulfated ions that were almost indistinguishable from each other. But Martini 3 actually has an attempt at reproducing Hofmeister series by parameterization that kind of looks like implicit Born ideas, but it's basically all translated into interaction strength. It fixes some of the biases in Martini 2, particularly for protein-protein interactions, and I'll show some examples for that. It's promising for protein-ligand interactions, which is a paper by Sejdiu in Nature Communications last year that tests a number of things. It's not in my slides or in the Martini 3 paper yet, but at an increased resolution and the bigger chemical space, you can actually look at more detailed processes, including protein-ligand interactions. And then just to spoil the party a little bit, it still has some of the major limitations from Martini 2, particularly entropy-driven effects. So one of the examples that I've always looked at or used is pore formation in bilayers. That's still tricky. Another blatant effect is taking 2 hydrophobic helices and calculating their interaction strength in water. Martini 2 gave exactly the right free energy, but the driving force was all enthalpy, 0 entropy. Whereas if you do this atomistically, which we did and published in PNAS, it's all entropy and almost no enthalpy. So Martini 3 still has that, and the smaller water particles don't really fix that. So some illustrations. So these are tests for dodecane at 3 different levels of resolution. So if you have these different levels of resolution, this should mix like ideal liquid and act like bulk dodecane, and it kind of does. And the same for water. There are 3 different water levels, 3 different water sizes, and you can actually mix them and they behave like a single liquid. And the paper has some more theory behind this and some more quantitative tests. But this is the general idea that you can have 3 different levels of resolution and make some parts of the system more detailed if you want to, and still already know beforehand that the interaction strength are more or less balanced, which wasn't true in the ad hoc smaller particles that we had before. This is another example, somewhat specific to a membrane protein. This is a toxin that forms a water channel. The blue line gives the number of water molecules going through this toxin with Martini 2. The red line, Martini 3. So the channel was basically not conducting for water at all in Martini 2, but it actually gives permeation that is comparable to atomistic with Martini 3. I think this is pretty remarkable given the simplified nature of water, but this relies on the smaller water particles. This is another example that I think is very important. This is protein-protein interactions, and it's become clear that Martini 2 makes proteins aggregate too much. This was true for many atomistic force fields as well, but we have not been able to sample this atomistically very well. With Martini, that's easy to sample, and basically if you have a solution of proteins, they all glue together. If you do this as a function of concentration, you can actually get different percentages of monomers, which you can do experimentally as well. And in Martini 2, you can get any concentration of monomers you want as long as it's 0, because they always aggregate. In Martini 2, Martini 3, there's a concentration dependence, salt concentration dependence of the percentage of monomers. So you actually can promote aggregation or deaggregation by adding salt. This is much closer to what it should be physically. Another example is transmembrane helices. So this has been a test system for a long time. We've published on this as well. And this is work from Robert Best. The Martini 2 had a massive well for bringing helices together. And we've published a paper on this as well, on looking at polyleucine and on Volt peptides. Basically, any two helices in Martini 2 wanted to be close together. If you look at the experimental values, and these are not that easy to interpret, but I think that's becoming clearer since we tried this 7-8 years ago. There's actually reasonable values now for association-free energies for dimerization. And Martini 3 gets exactly the right number for several different helices. So there's a sequence dependence to this, as it should be. And the structures look much closer to what they should be too. So Martini 2 is much too compact. Martini 3 structures compared to NMR for this particular dimer are actually pretty good. This is glycophorin. I don't have an example of this, but one of the things that bothered me with Martini 2 a lot, once you go to the complexity of the chemical review front page, all of these proteins have an exaggerated tendency to aggregate in Martini 2. And there are reasons for that that are non-trivial, but became apparent when looking at Martini 3. I think that will probably be much better in Martini 3, although we don't have sampling on these large systems yet. We're testing some of these things now. And I think that's going to be much better. And one difference is that Lennard-Jones density of proteins is different in Martini 3 compared to 2, by the way the mapping is done, by the way the interactions are localized. So some ongoing work. These are examples all from the Martini 3 paper currently in press in Nature Methods. I think that should appear in the next week or two. So with this, we can redesign the protein backbone. The examples I showed basically use Martini 2 topologies with Martini 3 parameters, but we can make more detailed protein backbones with more beats now that we have these smaller particles. You can also redesign lipids with more detail in backbone and head group. So we can actually distinguish better between different lipid types for lipid-protein interactions. And I think this will also help with getting phase diagrams for lipids correct. There's the potential for much better carbohydrates and nucleic acids. There is a carbohydrate force field, sweet Martini, that has certain capabilities, but it's not very capable of distinguishing between carbohydrates. With the higher resolution and with better electrostatics, that actually becomes possible. There are efforts in Theo John's group to make topology generation automatic and pre-parameterize all of chemical space. I think that's an interesting idea. In atomistic space, there's an almost infinite number of molecules. And Tristan Barreau has worked on this. But in Martini space, that space is much smaller. So in principle, you could pre-parameterize everything. And that would get away from people randomly parameterizing things for their own paper and randomly screwing up interactions or introducing inconsistencies in mapping, which I think was a big problem in Martini 2. And then there are further updated tools. Then it would be nice to implement Martini 3 in NAMD and VMD. Okay, maybe not us, maybe you can do that. I talked to several people about Martini 2 in the past, especially for NAMD. I think Martini 3 doesn't use anything new compared to Martini 2. It uses virtual sites pretty heavily. It still needs switch functions. And there's a bonded term that has coupled angles and bonds to avoid singularities in collinear beads. I think those are the main differences compared to what NAMD already has. Well, I know it has aspects of this already, but I think that's what you need to make Martini 3 run in NAMD. Okay, well, that's my story. I'd like to, well, I talked about a lot of different projects and a lot of different people. I'd like to single out Valentina Corradi and Estefania, who worked on the ABC transporters, BGP, and on the lipid-protein interactions initially. Bazian, who worked on GPCRs in the website, I spent some time on. Heidi and Williams Miranda in Sergei's group on the ceramides and Herc interactions. And then there are collaborations with a lot of people, Siwa John and a large group of people in Martini 3, Sergei Noskov and his group, and Hank Duff and his group for work on the Herc ion channels. And I should mention Dr. Hanif Khan, whose name I forgot to include on the slide, who did the MDFF simulations of Herc, and is also working on other EM structures. Thank you.
Thank you very much, Peter. Very nice collection of exciting developments. Congratulations on Martini 3 and the paper that is coming. We look forward to seeing more about it. So if anybody has questions, you can raise your hand. I'm supposed to see you, and then I'm going to call you to ask your questions. I can start, or you can put them in the Q&A. So let me see what we have before. Okay, so let me get started myself. Actually, I have a couple of questions. So is there any AI-based coarse grain model coming out of your effort? Have you been thinking along those lines or not?
AI?
Yes, I mean, you know, the force field sort of development sort of atomistically, so AI-based methods or force fields have been quite interesting and exciting. I was just wondering if in the coarse grain domain, you guys have been thinking along the same lines.
I haven't. Maybe other people have.
Okay. Fair enough.
Yeah, there is a roadmap at the moment for trying to improve the protein and lipid models. If there is any machine learning that would be helpful in that area, I think we'd be quite open to introducing that. I don't know how much you need that. Again, the chemical space for Martini is much smaller than atomistic. So in some ways that makes the problem easier to parameterize things. It's also easier to convert to things and get average properties that you use for parameterization, especially for lipids. If you look at Charm 36, there's a new paper by Ritz Buster on improving certain lipid types. But to look at mixtures and phase diagrams is almost impossible, and it will be almost impossible for quite a while. We had a paper in JAX earlier a year ago on mixtures, and it gets towards temperature dependence of phases, but it's nowhere near a phase diagram, and that's still only binary and tertiary mixtures. So the complexity of this atomistically is so complicated. It's so difficult to sample still. I think Martini's story is different in many ways. I don't know. I think it will be interesting to see. Anything that speeds up the development is useful. Because if you look at this, Charm has been around for a couple of decades. Even Martini has been around for 15 years or 17 years now. This is going way too slowly. It should be going much faster.
Hey,
Well, and then
Emad.
Yes, Alex, go ahead.
We're actually starting to do some tricks with using AI and deep learning to help facilitate the sampling of like parameter space in development and so on, as well as and then using it as in the context of like prediction of electrostatic parameters for like the polarizable force field, where you have to start to predict Tolle factors as charges as well as alphas and so on. So I've got some good people who are teaching me deep learning and so on. So we're starting to actually put it into practice, and hopefully we'll get some papers out over the next year. Great.
So we have expertise on this. Sergei Noskov actually has a large machine learning program in his group. It's addressing something else. It's addressing metabolomics, but we have a lot of machine learning expertise in our general environment at the moment. One reason I'm reluctant, I appreciate what Alex is saying for an atomistic force field, or even worse for the polarizable force field, where you have even more things to worry about. For Martini, the hope is still that you could basically enumerate all of the small groups based on organic chemistry and parameterize those, and then say this is transferable. That people have given up on that atomistically, and that was the idea behind the Drude oscillators. Oh no, this is so basic physics. We can do this at an atom by atom level, and it will automatically scale up to everything. But maybe at the coarse level, this might still work.
Mm-hmm. Okay, so let me actually go to the audience and see who has questions. So Alan K, you can actually ask your question if you turn on your microphone. I moved you to the panelists. Okay. So,
hi, thank you very much. I was curious as a non-MD guy, are you able to capture the ion selectivity with Martini 3? So distinguishing, say, between sodium and potassium.
So that's a tricky question. If you look at people working on potassium channels since 1998, so 23 years after KCSA structure came out, people are still fighting over the molecular basis for selectivity between potassium and sodium. That's something else that maybe depressed a few years ago as a complete lack of progress. So they're actually going to more and more complicated models because atomistic MD can't tell the difference. So Martini cannot actually address that at the level of potassium versus sodium selectivity in a selectivity filter in an ion channel. But if you're asking about the effect of sodium chloride versus potassium chloride on a lipid bilayer or on salting out proteins, there's more hope there. And for Martini 2, there was no hope because they were essentially the same. But that's no longer the case. So there's room for optimism there.
Okay, great. Can I slip in one other question? So did you say there were 3 kinds of water molecules?
So there's only one type of water molecule in nature, of course.
Yeah.
In atomistic models, that has an oxygen and 2 hydrogens, and the geometry is rigid. In Martini 2, the standard water model is a sphere that represents 4 water molecules. So when I said there are 3 now in Martini 3, there are now 3 spheres of different sizes, representing 4, 3, and 2 water molecules. So they don't look like a chemical water molecule in the sense that there's no oxygen and no hydrogens, but they do have the bulk properties and the sulfation properties in as much as possible within the approximations we're making of water, and they're comparable between the 2, 3, and 4 version. So you can mix them.
okay.
Sorry to jump in here. Thanks for the great talk. I was wondering, how does one choose which of the 3 to use then?
I don't know yet. So we've had a similar case. Like for Martini 2, we had 2 water models. The one I just described, basically the sphere, and a polarizable water model. And then QC made another one. So what we've done with those is, in cases where we think the polarizability is important, and this is not the electronic polarizability that Alex is talking about from his Drude oscillators, this is just the dipoles of the water molecules themselves. Where we think the polarizability is important, we would typically do 2 sets of simulations using both. And then we look at where the differences arise and whether that's important. So if you're looking at something like this fructotoxin pore I showed with water permeation, it seems plausible that the large water molecule model is not actually suitable for that level of detail. So there I would use the small one to begin with. But even the small one, even the 2 water molecules per beat is very much cheaper than atomistic simulations. So to do all 3 of them at the same time, if you want to, is not difficult. So I think what people will end up doing is you'll experiment with different levels of resolution, and you use higher or lower resolutions as controls in simulations, just like people these days are more likely to use multiple force fields before making wild claims about their protein folding or misfolding.
Got it, thanks. And is it released, Martini 3? Is it get out?
Yes, the parameters have been on the Martini website for quite a while for testing.
Oh, this is the Open 3 beta.
Yes.
Oh, okay, got it. Thank you. Great talk.
Thanks, Zoe. So let's move to Matthew Chauvin.
Hello.
Okay. Go ahead, Matthew.
Yeah, hi. So thanks for the talk. What I really enjoyed in Martini 2 was its simplicity, where you have a few beads and you can try and see if it's working or not. With Martini 3, as also mentioned Zoe, for Waters and Alan, it's a bit the complexity with new bead types, how we can choose in between the different beads in a way, and especially for lipids and so on. And so it may complexify a bit the way of other groups, more than Seward or you and well-known guys that are using Martini can use this type of force field. So
yes, that's an interesting point. We had a Martini workshop a year and a half ago, and Sejdiu is driving this towards more options and more complexity on the one hand. And I was arguing, well, if you make this too detailed, you're back to charm 36. But even the most detailed level now is much less detailed than charm 36 or an atomistic force field. And one of the things that I hope will help, and that's Sejdiu's plan, is to actually remove a lot of the choices that people can make. Like what I mentioned, if you can pre-parameterize chemical space, that means that if you have a new drug molecule, you are not free to pick your mapping. You are supposed to use the consistently derived mapping in the database of chemical space for that molecule. And what we've seen in a lot of applications in the literature, people map, typically overmap molecules. There are too many beats and too many interactions that make interactions too strong or unbalanced. So if you actually do a pre-parameterization of chemical space in a consistent way and say, well, if you want to use Martini and claim you're using Martini, you need to use this at this level of resolution or this level of resolution. You cannot fiddle with this beat to make it do what you want. People will still do that, and we've done that too. We've published papers on improvements to OPLS, where we fiddled with the way aromatic side chains interact with each other, but that never gets incorporated into real OPLS, and it's not an improvement people actually use. It points at a weakness in OPLS, or it did 20 years ago when we did this, but it's not actually sustainable in the sense that the changes we introduced were ad hoc. They fixed cation pi interactions for OPLS, but they weren't an improvement to the OPLS force field as a whole because we never tested it compared to all of the other components. So in that sense, it's not going to be different.
Yeah, okay. I was going to change the subject from Martini back to lipid-protein interaction. One problem actually we notice is that there are not really good experiments validating if we find a particular lipid-protein interaction or site or anything. So it's been, I don't know, over the last 10 years, I haven't seen much improvement or development in that respect. So what do you think? Is there any experimental kind of data that you guys use to validate or prove that this is the site where things find? You know, we used to have X-ray structures. Now with cryo-EM and the more natural membrane environment, things are a little bit better, but still there is nothing that we can really refer to. Look, this experiment completely validates our findings. What do you think, Peter? So has there been any change?
No, that's true. That is a weakness. And actually, I have a Canadian NIH equivalent grant due this weekend that talks about this. So I don't have the answer, and my secret ideas I'm not going to tell you. But basically, I think there is progress without any real answer at the moment. So there is progress, particularly in lipidomics and mass spec sample preparation. There is progress in single particle nanodisc cryo-EM rather than surfactants. And how you make those is a big deal. And the resolution is a big deal because in most cases you don't see the lipid nature, although you see lipid density. You can't identify it exactly, but this is getting better. And from what I understand from EM collaborators is that the theoretical resolution of EM is much higher than what people are doing at the moment, even though what they're doing is already quite spectacular. So there is room for improvement there. There's room for improvement in this mass spec. And one reason why we looked at these ion channels, well, one reason is Sergey has worked on these things and knows exactly which residues in HERG do what. But the main reason why I like them, and I generally don't like them very much, is because electrophysiology is super sensitive. So it won't actually tell you which residue, which lipid is binding, but any mutation for a binding site, if there is any lipid specificity, especially in reconstituted ion channels, that's a pretty clear indication that something is going on. It's not structural, but electrophysiology is spectacularly sensitive.
Yeah.
And
we have
reviewed
a
on
lot of papers exactly using that method, coupling it to electrophysiology, removing some residues, showing that it's lipid sensitive.
I think we both have an unfortunate interest in transporters, where all of the assays basically suck.
Yeah, that's right. That's right. Exactly. Well, thank you. So let's move. Miro, you can ask the last question. Miro Astor or Miro? I apologize for the.
Hi there. Sorry, I'm on a phone. Can everyone hear me?
Yep.
Yes, go ahead.
Yep. Cool. So I've been simulating the human phosphorylated CFTR channel for quite a while now. I'm a PhD student, and I'm over in Australia, so far away from all the big structural biology communities. I've just heard a lot of mistrust of this structure, and I've read the unfolding TM8 paper that you guys put out. I was just wondering, like, what makes everyone distrust this structure, because I don't really have a structural biology background.
Well, the main reason is that in general, unfolding a transmembrane helix inside the membrane is very expensive. So Steve White will be very upset just because you get hydrogen bonds that are not satisfied. So if you have a transmembrane segment that's unfolded, that normally requires other structures close to it to stabilize that unfolded state, even if it's a local unfolded state. The TM8 is kind of special because it's not actually as central, and it's kind of close to the interface with lipids. And we argued that, yeah, it might be stabilized by water defects, but if it does, then perhaps it's also a drug binding site. But in general, people just energetically don't believe that transmembrane helices are unfolded unless you can see what unfolds it. So if there are salt bridges or hydrogen bond networks in the rest of the protein that stabilize the unfolded state, which is not the case for TM8. TM8 is lipid exposed. And to have it unfolded in the location it's unfolded in the structure suggests that there's a water defect that I think has a pretty high cost. JC has worked on this as well. I wonder what he thinks.
Yeah, I just was going to chime in on that. So I also had my doubts about it, but one of Zhu Chen's more recent structures shows ivacaftor binds there. Yeah. So like you said, maybe it's a drug binding site, although still why it happens in the first place, I wonder.
So we're leaning in the same direction. We've done a little bit of follow-up on it, looking at a drug binding potentially. It might be a special site, and then drug binding is one way to indicate it actually is a special site. But people have not really been able to link it to a function. But CFTR is annoying because it's an ion channel. So in a way, it has the same sensitivity as an ion channel, but it has all of the problems of a transporter with undefined states that are slow. And so as ion channels go, it's annoying compared to potassium channels. And the mechanism people ascribe to CFTR are just insane with 27 different states and transitions. And I don't believe any of that. Never mind. That's very difficult to interpret.
My supervisor says the same thing. Thank you.
Sure. Okay, so the very last question, Kiana. Kiana RP. Okay, so there's a question. Kiana is wondering about adding accelerated MD or targeted MD to Martini. Is it possible?
Sure. There's nothing. Well, accelerated MD is a little bit different. So accelerated MD fiddles with dihedral transition potential barriers, which in Martini are not actually all that important because proteins are not flexible, generally speaking. We don't reproduce internal conformational dynamics of proteins. And in lipids, you don't, or in loops, you don't actually get barriers that you need to cross. So accelerated MD makes less sense unless you find some other potential term you want to boost. Targeted MD works the same way as in atomistic. In Chromax, you can just apply restraints to, and same in MD, you can just apply restraints in the same way as you do atomistically. All of the normal free energy things work as well. We routinely do FEP and umbrella sampling type things.
Absolutely. Okay, so I need to sort of stop it. Peter is meeting with someone.